Global warming has been a problem over recent decades affecting the environment worldwide. Mexico is very vulnerable to the effects of this phenomena as it has been reported that over the last 50 years the temperatures have increased by about 0.85 degrees Celsius. Our region, the northeastern part of Mexico, is characterized by longer periods of heat in comparison to the rest of the country. Not only that, but there has been an increment of incidence of cutaneous malignancies as it has been reported since 2024. Our government has established recommended precautions against these conditions, including protective clothing, reducing sun exposure, and most importantly, applying sunscreen. It is common that once applied, the sunscreen is deposited into water due to contact with the skin, resulting in a deteriorating ecosystem. As it has been shown, its UV filters can cause coral bleaching by affecting the zooxanthellae light in symbiosis. In order to provide sunscreen that does not harm coral reefs, the UV filters used need to be changed for alternative biocompatible components that serve the same function without impacting the flora and fauna of any body of water. For example, pyridine 2,6-dicarboxylic acid or dipicolinic acid (DPA) is a constituent of bacterial endospores, produced during the sporulation phase as a secondary metabolite of the lysine pathway of some species of the genera Bacillus and Clostridium. For these organisms, DPA plays a key role in providing heat and UV protection to their spores through calcium chelation. Due to its protection against UV radiation and biodegradability properties, DPA could be potentially used as the active ingredient in sunscreen, replacing the common UV filters. To use DPA as a replacement, it is necessary to ensure gram-scale production. Previous work has yielded promising results through strain and metabolic engineering using E. coli and substrate engineering in B. subtilis. However, there has yet to exist a proposal implementing genetic and metabolic engineering of Bacillus subtilis to efficiently control the production of DPA during the stationary phase, which is the phase where the highest DPA yield can be obtained, as we identify as part of our study analysis. These strategies facilitate better monitoring of potential yield during industrial scalability and reduce costs that could be attributed to complex culture media during the bioprocess. Our project, DPAHelix, aims to use a genetically modified strain of Bacillus subtilis as a chassis for the overexpression of the gene SPOBF using a stationary phase-specific promoter and identify potential metabolic gene knockouts to improve substrate flux to DPA production. Our proposal also covers the analysis of the DPA produced as a component of a sunscreen through a chemical and biocompatibility analysis. Under this computational approach, we expect to establish a strong foundation for characterizing the chassis for future implementation in the laboratory, followed by industrial scaling and ultimately the creation of a formulation tailored to consumer needs. This proposed sunscreen will therefore be an eco-friendly alternative that maintains its properties under UV light and reduces damage to flora and fauna in bodies of water. As a team, we structured the project and, after several in-person meetings in the lab, we finalized the main points of our project. Number 1. Identification of the metabolic flux toward the DPA precursor under glutamate and glucose media using genome-scale metabolic models. Number 2. Analysis at the transcriptional, translational, and enzyme kinetic levels and bioreactor level potential production using the stationary phase-specific promoter to the SPOBF gene, which encodes the main enzyme dipicolinate synthase. Number 3. Performing an enzyme engineering analysis to improve the catalytic activity of dipicolinate synthase. And number 4, identifying the physicochemical and biocompatibility characteristics of DPA as a potential active ingredient in sunscreens. This project was realized under the supervision of our principal investigator, Dr. José Claudio Moreno Rocha, our advisor, LBG Mauricio Reyes Elizondo, and finally, our advisor, LBG Ulises Janet García Cepeda.
We use a genomic-scale metabolic model of Bacillus subtilis 168 obtained from the Big Models database to evaluate DPA production using glutamate as a carbon source. All analyses were performed in Python using the CoraPy library. Flux balance analysis was applied under the steady-state assumption for intracellular metabolites. The objective function was defined as the maximization of DPA production by directly optimizing the reaction corresponding to its synthesis within the metabolic network. This approach allows us to evaluate the theoretical metabolic potential of the system rather than a specific experimental condition. To assess the role of the carbon source, glutamate uptake was enabled as the main carbon input, and simulations were compared with glucose under equivalent uptake constraints. In addition, based on previous studies, we evaluated perturbations of the acetylene biosynthesis pathway through knockout and knockdown strategies to determine whether redirecting carbon flux could enhance DPA production. Finally, flux distributions, reaction activities, and shadow prices associated with the objective function were extracted and analyzed to identify key pathways and metabolic constraints influencing DPA biosynthesis.
Enzymatic engineering of the picolinate synthase to improve catalytic activity. To increase the concentration of DPA, we propose improving the catalytic activity of the picolinate synthase. We evaluated databases such as Brenda, Uniprot, RIA, IUBMB, and PDB to gather enzymatic data. However, the lack of catalytic information shifted our objective toward identifying conserved domains and proposing a list of amino acids related to the active site. We selected 50 amino acid sequences homologous to B subtilis picolinate synthase subunit A using the NCVI FASTA program. Then a multiple sequence alignment was generated using MEGA. Finally, using the Interpro program, 10 representative sequences were analyzed to observe the domains comprising the enzyme. This process was repeated for the subunit B. To predict the three-dimensional structure, we utilized algorithms from the Tamilum Bio platform, such as AlphaFold, Boltz2, Chea1, AFsample2, and ESMFold. Since we were unable to obtain a high-confidence AB complex, we decided to predict the structures of the subunits separately. We performed molecular docking to the precursor HTPA using ESMFold, Autodocbina, and DiffDock to quantity binding affinities. The software ProLift was used to validate the binding results of Autodocbina and identify specific amino acids that interact with the precursor in each subunit.
Based on previous reported enhanced DPA production using glutamate, we first compared glutamate and glucose as carbon sources using flux balance analysis. As shown, glutamate consistently superior higher DPA fluxes across all evaluated growth rates. With glutamate, DPA production reached approximately 6.8 millimole per gram of dry cell weight per hour at zero growth rate, and gradually decreased with increasing growth. In contrast, glucose superior simulation showed lower DPA fluxes and did not reach high growth rates. Although precursor to product conversion was efficient in both cases, glucose metabolism exhibited a pronounced diversion of carbon toward acetylene synthesis, particularly at intermediate growth rates. This behavior is consistent with previous studies showing that high glycolytic flux leads to excess pyruvate, which preferentially activates the acetylene pathway and competes with alternative biosynthetic routes such as DPA production. To evaluate whether acetylene biosynthesis limits DPA production, we analyzed genetic perturbations of the acetylene pathway. Complete deletion of the LSSD gene cluster proved lethal, indicating that this pathway plays an essential regulatory role in central metabolism. Therefore, a knockdown strategy was implemented to partially reduce acetylene flux while maintaining cellular viability and redox balance. Under glucose conditions, the knockdown effectively reduced flux toward acetylene. However, DPA production remained unchanged compared to the wild-type strain. This indicates that acetylene biosynthesis is not a limiting factor for DPA production under glycolytic conditions. When glutamate was used as a carbon source, acetylene flux was already low both in the wild-type and knockdown strains, and no changes in DPA flux were observed. These results suggest that under non-glycolytic conditions, acetylene biosynthesis plays a secondary role in carbon redistribution and does not directly compete with DPA synthesis. Analysis of central metabolic fluxes revealed clear differences between the two carbon sources. Under glucose conditions, metabolism was dominated by glycolysis, the pentose phosphate pathway, and energy-generating reactions, while fluxes toward the aspartate pathway, precursor to DPA, remain low. This agrees with previous transcriptomic and fluxomic studies showing that glucose metabolism prioritizes energy generation over biosynthesis. In contrast, glutamate entered directly into the Krebs cycle, promoting oxaloacetate and aspartate formation and providing a more direct and efficient route toward DPA biosynthesis while maintaining adequate energy production. Shadow price analysis further showed that metabolic constraints depend strongly on the carbon source. Under glucose conditions, DPA production was mainly limited by structural and redox-related metabolites, whereas under glutamate conditions, limitations shifted toward cofactors and nitrogen-related intermediates. Importantly, metabolites associated with the acetylene pathway did not emerge as limiting or inhibitory factors in any scenario, reinforcing the conclusion that acetylene biosynthesis is not a metabolic bottleneck for DPA production.
Alright, now let's talk about the genetic circuits and the mathematical models that we use to predict the protein synthesis and product accumulation. We use different models for predicting critical aspects from DNA transcription to DPA synthesis. We analyze values like transcriptional kinetics, translation dynamics, enzyme catalysis, and product accumulation across four constitutive promoters under varying metabolic conditions and different substrates. First, we calculated three different parameters for four different promoters: molar transcription rate, transcripts per second, and steady-state mRNA. Steady-state mRNA is quite important since is the number that represents the equilibrium between production and degradation of mRNA concentration. Hence, when we take over this analysis, we reach a number of 2183 with promoted GLB, which is a stationary phase promoter, which was 8.2 times higher than almost any other promoter. So PLB in this first state was our best option to choose since it has a very high steady state parameter. So hence this could be related to a high protein accumulation, thus leading to a high product production. Now in these models we predict the mRNA concentration in 1 hour and the SPOVF enzyme production in 24 hours. On both scenarios, promoter GLB surpassed by far all the other promoters, producing almost 10 times the amount of mRNA concentration and SPOVF enzyme. Next, we use the Michaelis-Menten kinetics to model the enzymatic velocity of SPOVF under two metabolic flux scenarios. In this model, we use glucose and glutamate. In this model also we can see that PLB was the highest catalytic rate compared to the other constructs. Under glutamate supplementation, we reach a max velocity of 1.71 micromole per second, this being the highest above all the other promoters and even above the glucose supplementation. Finally, we model the product accumulation over a time of 24 hours in Bacillus subtilis. In our initial scenario, we use a specific metabolic flux of glucose and another of glutamate. The results were that the best promoter was again PYLB with a flux of glutamate, producing 1.39 grams per liter. This result was lower than we expected since we expected 30 grams per liter. So we decided to change the flux rates using 5, 15 and 25 grams per liter. In these scenarios, we use only two promoters, the two highest in the last results, P43 and PYLB. The results were that only the promoter of PYLB, which using the substrates of glutamate with 25 grams per liter, achieved a production of 33.27 grams per liter of DPA, achieving the desired DPA production of 30 grams per liter.
The picolinate synthase is exclusively found in Bacillus and Clostridium genera, with only a few reports in fungi. Importantly, the enzyme structures and the metabolic pathways involved vary significantly between genera. This variability limits how far evolutionary computational analysis can be applied, and its subsequent specific identification in the EC classification. However, a previous work demonstrated that both subunits independently synthesize DPA, suggesting that the catalytic activities of the subunits can be characterized individually. Based on this, we use AlphaFold to predict the three-dimensional structure of each subunit, with a high confidence score of 96.56 of subunit A and 92.95% of subunit B. Among the docking algorithms tested, Autodoc Bina provided the most reliable results. The results from this algorithm were the ones where ProLief identified the most interaction. Binding affinities of -6.593 and -5.338 kilocalories per mole were recorded for subunits A and B respectively and validated using ProLief. The predicted binding regions align with those identified by Interpro, particularly for subunit A, where the active site is located within a specific functional domain. Notably, we observe stronger interactions in subunit A compared to subunit B. This corresponds with previous findings, who reported that subunit A produces higher levels of DPA on its own, while the combined A-B complex yields the highest overall concentration. Although our computational results show that both subunits can function independently, it remains possible that the assembly of the full complex optimizes DPA production. Previous studies of enzymes in the same subclass with an EC number of 1.3.1 indicate that active sites are often shared at the interface of two subunits. This suggests that dipicolinate synthase might undergo similar folding to join the two identified active sites. These results provide a computational foundation for further study of dipicolinate synthase catalytic activity.
To establish the formulation of the UV filter, PubChem database was used, from which the qualitative data such as structure and functional groups, as well as quantitative data including the donor and recipient electrons, partitioning coefficients, and additional elements were obtained. For this analysis, a hosted Jupyter Notebook service named Google Colab was used. The analyses were implemented there using RDKit, Matplotlib, and NumPy libraries.
Now, focusing on the partition effect of DPA and its physicochemical properties, they are important because they will define which path we'll take for our formulation design. We evaluated the chemical characteristics of DPA at a pH close to that of human skin. This analysis helped us predict whether the most suitable solvent system for the formulation will be polar or nonpolar. The results show that DPA has a negative partition coefficient. This indicates that the compound has a higher concentration in the aqueous phase rather than in the oil phase. In other words, DPA is more polar and has low lipophilicity. Based on this result, an oil in water or OW emulsion system was selected. In this type of system, oil droplets are dispersed within a continuous water phase. An OW emulsion naturally forms two phases, a polar phase and a non-polar phase, creating surface tension between them. To achieve a stable emulsion, physical force is applied using agitators, which generate microeddies. These microeddies break the interface between the oil and water phases, allowing the emulsion to form. Heat-sensitive components such as fragrances, humectants, and fixatives can be added in a final step, reducing the risk of degradation. As our intended product will be applied on skin, serious health and biosafety concerns arise. For this reason, we made a series of in silico analyses that could indicate us if DPA is safe to use in the conditions implied. General safety studies were run using online drug design software such as SwissADME and Protox 3.0, which showed how physicochemical properties of DPA relate to its drug likeness and its probable toxicity respectively. Results from these were thoroughly analyzed after obtained using R scripts made by us and visualizing data in predictions from Swiss ADME indicated our UV filter to be potentially water-soluble, with low skin permeability, no inhibitory interactions with cytochromes, nor blood-brain barrier permeability, characteristics predominantly present on traditional compounds. Also, we found using Protox 3.0 low probability for potential nephrotoxicity, no ecotoxicity, and chemically induced blood-brain barrier toxicity. The latter shows controversy between models, leading to a need for future research in order to establish certainty. On the same line, mutagenic analysis was made using Boltz-2 via the Tamarind Bio platform. We performed a biomolecular simulation of interaction between DPA and DNA for us to discard if it could have an intercalating activity. 4 of the 5 simulations done had a high confidence score, from which all had a low interface predicted TM score, implying a low binding probability and therefore an unlikely role as an intercalating agent. Now, related with the immunogenicity, a blind molecular docking simulation using autolock BINA was executed to unveil the interaction between DPA and a panel of receptors related to the innate immune response, principally for the detection of pathogen-associated molecular patterns such as TLR1, TLR2, CD14, and MBL, as well as the osmotic stress receptor TRPA1. According to the results obtained, all proteins tested could interact with DPA having a mean affinity value of negative 4.95 kilocalories per mole. Unfortunately, we think the outputs of this analysis could be biased or false positives, so a cross-examination with experts in disciplines such as physical chemistry, immunology, and analytical chemistry is necessary to corroborate our findings. Finally, in order to study the possible antimicrobial effect of DPA on the skin microbiome, 7 clades of bacteria present on the moist, dry, and sebaceous sides of the human skin were selected, as well as a genus of fungi named Malassezia. A representative microorganism was chosen for each clade, and the essential proteins' 3D structures were downloaded through PDV. Then code was developed to evaluate the interaction between ligands and DPA using a molecular docking-based. Unfortunately, due to inconsistencies in data processing and a lack of information on databases, the results could not be confirmed, and a more accurate methodology must be used to define the impact of DPA on the skin microbiome. Nonetheless, the antimicrobial activity of DPA in gram-negative bacteria has been previously reported, establishing the possibility of providing UV protection with dual activity for patients with skin conditions. Additionally, it has been reported that DPA possesses insecticidal activity towards Hemiptera, an order of insects, including the whitefly.
Throughout the in silico simulations, we achieve high yieldings of DPA productions, where we obtain through the use of metabolic engineering, mathematical modeling, and substrate optimization. We also like to add that we managed to identify specific amino acids that could potentially be involved in the catalytic activity of DPA synthase, thus having a positive effect in producing a larger amount of DPA. These results could potentially show that computational approaches in this study and the development of new technological solutions are possible, and we need further examinations and further studies to continue these investigations.